Adolescence is an especially important period for the question that I'm asking because the systems involved in emotional reactivity and regulation are still developing. Totem and Gavin, 2016, described adolescence as a period of substantial reorganization in amygdala prefrontal cortex circuitry. But at the same time, adolescents routinely encounter salient academic stressors, including timed assessments and performance evaluation. So the question becomes, can we change how adolescents respond to one of those stressors by changing the way they think about their ability to move forward? Importantly, I am not defining hope as optimism or simply believing everything will work out. Snyder's hope theory defines hope through two interacting components, agency and pathways. Agency is essentially the will. I believe I can continue toward this goal, and pathways are the ways. If this route is blocked, I can generate another one. That distinction matters. A student can desperately want an A, but wanting an A is not hope. Hope requires both perceived capacity to act and the ability to generate routes around obstacles. Now we move from psychology into neuroscience. Wang et al. Found in 2017 that dispositional hope statistically mediated the relationship between spontaneous medial orbital frontal cortex activity and anxiety. The MOFC is involved in evaluation and contextual appraisal, while the amygdala helps process emotional significance and salience. But there is an important limitation that research does not establish that increasing hope causes an immediate neural response to stress. That distinction is what led to this proposed experiment. We have behavioral research connecting hope with academic functioning and adjustment. We have intervention research suggesting that agency and pathways can be taught. And we have neuroimaging research identifying neural correlates of dispositional hope. What we don't know yet is whether experimentally increasing an adolescent's state hope immediately before academic stress changes their psychological response and the coordination between the MOFC and the amygdala during that stressor. That is the bridge this study is designed to test, as you can see below. To test the question in this experiment, I propose a randomized active control design. The independent variable is condition. Adolescents receive either a 20-minute hope intervention or a 20-minute active control activity. They then complete the same academic stress task. The behavioral outcomes are state hope and state anxiety. The primary neural outcome is task modulated functional connectivity between the medial orbital frontal cortex and bilateral amygdala. The active control is important because it helps isolate the effect of the hope content rather than simply receiving attention or completing an activity. An example of this is in the slide in front of you. I have 4 hypotheses. First, adolescents receiving the hope intervention should demonstrate a greater increase in state hope. Second, they should report lower state anxiety following the academic stressor. Third, I predict a different pattern of MOFC amygdala connectivity between conditions. Notice that this hypothesis is intentionally non-directional. Developmental neuroscience does not support the simplistic assumption that more or less connectivity is automatically better. Finally, across participants, greater increases in state hope should correlate with lower post-stressor anxiety. The target sample in this experiment is 120 adolescents between 14 and 17 years old, recruited from a public secondary school in the Mid-Atlantic region. Participants will be randomly assigned 1-to-1 with 60 in each condition, and randomization will be stratified by age group and gender. Because this involves minors and MRI, safety is central. Participants must meet MRI eligibility criteria, and the study requires both parental consent and adolescent assent. The proposed imaging protocol uses a 3 Tesla MRI scanner with MRI-compatible visual presentation and response equipment. Psychologically, state hope will be measured using Snyder et al.'s 6-item state hope scale. State anxiety will be measured using the state anxiety subscale of the STAC. During imaging, Participants complete a timed verbal reasoning task using age-appropriate reading, inference, vocabulary, and evidence selection items. This gives us a controlled academic stressor rather than trying to recreate stress artificially outside an educational context. This slide shows one of the most important methodological features of this whole study. Both groups received 20 minutes comparable reading and writing demands, facilitator attention, and encouragement. But only the HOPE group identifies a meaningful academic goal, generates multiple pathways, anticipates obstacles, identifies supports, and commits to a concrete next step within 48 hours. The control group completes neutral school-related reading and categorization activities without goal-setting, pathway generation, agency statements, or future success visualization. So if the groups differ afterward, we have a stronger basis for attributing that difference specifically to the HOPE intervention. Here is the procedure for the study from beginning to end. First, families will complete eligibility screening, informed consent, and adolescent assent, and MRI safety screening. Second, participants complete demographic measures, baseline state HOPE, and state anxiety measures. Third, concealed randomization assigns them to either the 20-minute HOPE intervention or active control. Immediately afterward, HOPE and anxiety are measured again. That gives us the first critical comparison. Did the intervention actually change state HOPE before the stressor occurred? Participants then enter the scanner. After practice trials and anatomical imaging, they complete a 12-minute timed academic stress task with bold fMRI data, which is collected. They know both speed and accuracy are being measured, creating a standardized performance evaluation context. After leaving the scanner, participants complete the state HOPE scale and stack for a third time, along with perceived stress and effort measures. Finally, they receive a standardized debriefing, and control participants are offered the HOPE activity. Behavioral data will be analyzed using mixed effects models examining condition, time, and their interaction. For the neural outcome, task-related MOFC amygdala connectivity will be estimated using psychophysiological interaction analysis with per-specified regions of interest and rigorous motion quality controls. And I want to emphasize an important limitation in interpretation. Functional connectivity tells us about coordination of neural activity, not causal influence between brain regions. Ultimately, this study asks a fairly simple question with potentially important developmental implications. If we give adolescents more perceived ways forward, does that change what happens when they encounter an obstacle? Here are my references.